Okay, well, good morning. Welcome back. Today's lecture will be about the gastrointestinal tract as a microbial habitat. So we'll start talking about the microbes themselves beginning next lecture. This lecture is about setting the stage for the microbes. Okay, so we have several objectives. You should learn several things by the end of the lecture. You should be able to identify characteristics in the gastrointestinal habitat that's important to microbial growth. You should be able to identify gastrointestinal anatomy of different mammals and its relationship to diet and microbial fermentation. And then finally, to distinguish between different types of herbivory and the merits of each type, because in herbivores, microbial fermentation is important. Okay, so you have reading from one chapter in the textbook, the Russell textbook. It's chapter 6. And just as a general note, what is most important is what you'll see in lecture. The textbook exists to help supplement lecture so you can better understand the concepts in lecture, but the material that you're going to be tested over is that in the lecture. If it's in the lecture, you need to know it. If it's in the textbook but not in the lecture, you don't need to know it. Okay, so the gastrointestinal tract is the site of diet digestion and microbial fermentation. And we have a familiar example of the sheep here with the rumen. Just to briefly go over the kinds of microbes that you can find in the gastrointestinal tract, we have bacteria, protozoa, fungi, methanogens, and viruses. We'll talk about these next lecture. We're just talking about the habitat, the gastrointestinal habitat in which these microbes reside for this lecture. Okay, so what in the gastrointestinal tract allows microbes to grow? There are several important characteristics. One is warm temperature. Body temperature is between 38 to 41 degrees in most species, and microbes need these warm temperatures in order to grow rapidly. Another feature is near neutral pH. 5.5 to 6.9 are typical pH values in the gastrointestinal tract. And again, gastrointestinal microbes, they're adapted to these pHs. Microbes need things to grow. They need substrates and growth factors. And there's a good supply of these in the gastrointestinal tract. Carbohydrates provide energy and carbon. Amino acids and ammonia provide nitrogen. And we have minerals, vitamins, and other growth factors. And we'll talk about what those other growth factors can be, but the major ones are carbohydrates, amino acids, ammonia, minerals, and vitamins. And another feature that's important in the gastrointestinal tract is no oxygen. Gastrointestinal microbes are killed by even trace amounts of oxygen. They require a no oxygen or an anaerobic habitat. And now that we've gone over those characteristics, I guess I'd like to ask you, why do you think the human stomach pH is so low? We said that we have a near neutral pH in the gastrointestinal tract. Why in the human stomach is the pH so low?
Do we just speak out?
Yeah, you can just speak out.
I think it's low because one, it's mainly for protein digestion, to break down protein into amino acids easier. Another reason is to prevent pathogens going into the intestine and destroy the flora of the intestine.
Yeah, so there are two important things. One is for protein digestion. So our stomach has adapted, or we've adapted to having low pH in our stomach. Protein is begins digestion at low pHs. The other is to prevent microbial growth. So growth of pathogens and really all microbes in the stomach. We don't want microbes breaking down our diet before we've had the opportunity to digest them. So when I'm talking about characteristics important to microbial growth, it's for parts of the gastrointestinal tract where we want microbes to grow. We don't want them to grow in the stomach, and that's why the pH is so low. So all these characteristics, it's just for the parts of the gastrointestinal tract where we see microbial growth, okay? So now we'll look at different parts of we'll look at different gastrointestinal tracts, and we'll look at their anatomy. We'll look at 3 different groups of mammals. We'll look at carnivores, insectivores, and frugivores. We can lump all of those together. We'll look at omnivores separately, and we'll look at several kinds of herbivores. And what we'll see is that microbial fermentation becomes progressively more important as we go down. So it's not that important carnivores, insectivores, and frugivores. It becomes more important in omnivores, and it's very important in herbivores. Okay, so what we're going to see is several gastrointestinal tracts. You've seen gastrointestinal tracts before, so we're going to assume that you can identify some parts on your own, that you can identify the stomach, the small intestine, the cecum, and the colon. We may point them out, but even if we don't, you need to know where they are. So if they're not clear, just speak up, and we can point out where they are. And there will be other structures that you'll need to identify that will be specifically pointed out to you. Okay, so this is the gastrointestinal tract of the human. We'll now see the gastrointestinal tracts of carnivores, insectivores, and frugivores. So we have the cat as a carnivore. We have the insectivorous bat as an insectivore, and a squirrel monkey as a frugivore and insectivore. So carnivore, we know that that means that the animals eat meat. Insectivore, if you haven't heard that before, you already know what it means. It eats insects. Frugivore, they eat fruit. And I wasn't able to find an animal that ate just fruit, so we get a squirrel monkey, which eats both fruits and insects. Okay, so in these animals, there's little microbial fermentation. We have a simple glandular stomach in all 3. They look a lot like our own stomach. Another feature is that they have a short hindgut. And sometimes it's indistinguishable from the midgut. So in the cat, you can see a hindgut. It is distinguishable. In the insectivorous bat, it's kind of hard to see where the midgut ends and where the hindgut begins. And in the squirrel monkey, it has this cecum here. Otherwise, it doesn't have much of a hindgut. And the reason that there's no complex architecture is because the diet is digested very quickly. So a cat that's eating meat, its diet is not hard to break down. It needs to break down the protein, fat, and once it doesn't need any microbial fermentation to help in that process. In fact, it would be bad for the animal if microbes would digest the protein and the fat. Insects are also easy to digest, and fruits are extremely easy to digest. So we see very little evidence of the need for microbial fermentation in these, and the gastrointestinal tract reflects that. Okay?
I have a question on this.
Yes, definitely. Please break in, or it's going to be a boring lecture. Yeah.
Yeah. So when you say complex architecture, what do you mean by that? Are you talking about like organs, or is that like you see less villi, or what do you mean by that?
So it'll probably be a little bit clearer when we go to the next slides. Architecture, I mean organs. Like this is a simple stomach. This is not a rumen.
Okay.
So it's very simple. You have simple stomachs. You have hindguts and midguts that are not large. They don't have any elaboration. Okay. So, and just to skip ahead a little bit, think about the rumen. Think about the different compartments of the rumen, which are made to retain, digest, and microbes.
Okay, cool.
Is that clear?
Yep, better.
Okay, yeah, definitely jump in when you have questions.
So is the surface area lower in these digestive tracts, or like in relation to the size of the animal, or is that unrelated?
So my first answer is I don't know exactly. They have to maintain high surface area to absorb, like villi in the small intestine need to maintain high surface area to absorb nutrients. But the gastrointestinal tracts are smaller in comparison to the animal. So I don't know exactly. I don't know what the balance of factors would be. So, I mean, you have, as I'm talking through it, you have high surface area in the small intestine, but you have relatively small surface area in the stomach just because it's smaller than in herbivores.
So the surface area isn't related to the complex architecture.
No, I wasn't really referring to surface area. So it refers to the size of the organs and also how compartmentalized they are. So like the rumen is really big and it also has separate compartments, and those help slow the passage of digesta.
Great, thanks.
Yeah. More questions for clarification? This is very good. Okay, we'll move to the next slide. Again, stop me at any point. Okay, so the next class of animals that we'll discuss are the omnivores, which eat a diet of meat and plants. They have some microbial fermentation. We see in the black bear and the possum a simple stomach. So that looks a lot like the mammals that we just saw. But now we see a more complex gastrointestinal tract as we go down. The midgut and the hindgut are a variable length. The midgut is really, really long in the black bear. And in the possum, the hindgut is large. So it's often better developed or longer than in the carnivores, insectivores, and frugivores. And the reason for the longer and more complex tract is that the diet is more difficult to digest. So plant material is difficult to digest. It must be fermented. So in the possum, we have the large hindgut in order to retain microbes and to permit fermentation of material there.
Sorry, could you repeat that about the possum?
Sure. So in the possum, we have a very large hindgut. And the size of the hindgut allows plant material to be retained there and for microbial fermentation to take place there. So the best, yeah, go ahead.
Yeah, so I don't know, thinking about back to the carnivores, and you were saying there's little microbial fermentation. So the fermentation is important for the digestion of plant material. But in the case of a carnivore that's eating protein, is that just the, like the cat, that's just the host enzymes are breaking down the protein? Or is there another process that microbes play in carnivores that they're not, it's not fermentation, it's something else? I don't know.
So there is very little microbial fermentation. I don't want to tell you no microbial fermentation at all, but there's very little microbial fermentation. The host is breaking down the proteins. So the digestion in carnivores is like in us. Protein digestion begins in the stomach and continues in the small intestine, and the proteins are absorbed in the small intestine. I guess one way of looking at it is if cats had microbial fermentation, they would not be able to digest and absorb the protein directly, even though they're capable of doing it. And they would lose the amino acids and some energy in that protein. So it's detrimental if there's microbial fermentation in their tract.
Oh, okay.
Can I ask a question?
Yeah, go ahead.
As you know that some humans don't eat meat products, and do they have like microbial fermentation? And do we have like microbial fermentation in our guts, or?
We definitely do. Yeah. So let's go back. So you can see that we have a decent sized cecum and colon. So our tract is that of an omnivore. Fermentation takes place, and about 10% of our energy requirements are met by short-chain fatty acids produced in the cecum and colon. So 90% comes from our diet being digested and absorbed by us, and then 10% comes from fermentation of fiber and other plant material that resisted digestion upstream. 10% of that comes from fermentation of that material and our absorption of the short-chain fatty acids. So you mentioned that there are some people who don't eat animal products.
Yeah.
I would presume in those people that the energy requirements are microbes probably provide more than 10% of the energy requirements, but I don't know for certain.
Okay, thank you.
Yeah. Okay. So we have carnivores and insectivores and frugivores where we have very little microbial fermentation. I don't want to say none, but very little. We have the omnivores where we have more microbial fermentation, and we see that the tracts become larger and they become more complex, and that's to help retain the digesta and microbes to promote fermentation. And now we're going to go to the herbivores. And we have several types of herbivores, but in all of these, microbial fermentation is important. So we have foregut fermenters, which include the ruminants, which we're probably most familiar with, but also non-ruminants. We have hindgut fermenters, like the horses, and we can break those up into cecal and colonic and cecal colonic fermenters. This is just a preview of what we're going to see. We're going to see tracts from each 4 of these. And we're also going to talk about not only the similarities between these different ways of digesting plant material, but also the differences and how they're important to the animal. Okay, so let's begin with the familiar example, the ruminant foregut fermenters. They have an enlarged forestomach. So here's the sheep again, and here is an antelope. And we can see the rumen is, or the forestomach in general is large in each of these. And this enlarged forestomach, we know, is the site of digester retention and microbial fermentation. And that's where most of the diet is fermented. We should also point out that there is a small to mid-sized cecum in these, where we have some additional fermentation. So we call these foregut fermenters. Most of their fermentation occurs in the foregut, but some occurs in the hindgut too. So it's kind of a spectrum. They're just foregut fermenting leaning. Okay, and so to break down what we have in the forestomach exactly, we have the 4 compartments. We have rumen, which is involved in particle retention and microbial fermentation. The reticulum, it's the same as the rumen. The omasum, we really don't know the primary function of the omasum, but it's probably involved in particle retention and water absorption. And the abomasum is a glandular stomach. So it serves the same function as in our stomach. And you can see these compartments in different ruminants. In the goat, for example, we have the rumen, the reticulum, the omasum and abomasum. So this will be familiar to many of you. For those who, this is old news. I just want to point out a piece of trivia, which is not all ruminants have these 4 compartments. In the chevrotain, the omasum is absent. So the forestomach of the chevrotain is shown here, and sure enough, there is no C. There is no omasum. It's lacking, and we can see a chevrotain pictured here. It's an interesting animal. It's about the size of a rabbit. It resembles the primitive ruminant, the ruminant that first evolved 55 million years ago, probably more than any other ruminant today. It eats fruits and some other plant material, but if it sees an insect or a mouse, it will eat that as well. So they're again some interesting ruminants.
Sorry, can I ask a question?
Yeah.
What do you mean by the particle retention?
Particle retention. Good.
Yeah.
So particle retention refers to keeping the particles in the forestomach for an extended time. So when the cow ingests food, particles are retained in the forestomach, in the rumens and reticulum specifically, for about 24 hours, 24 to 48 hours. Does that make sense?
Yeah.
Yeah. So in our own stomach, I'd have to look it up, but I think they're retained in food is retained in our own stomach for only a few hours. So the rumen and the reticulum, it is designed to retain particles for a long time, and that's simply so that microbes have an opportunity to ferment them. Microbial fermentation is a slow process. It doesn't happen in just a couple of hours. It takes an extended period, and so the rumen and reticulum are designed to retain the particles and to ferment them. Good question.
Where is the chevrotain native to?
The chevrotain is native to Asia. I think that there are some also distributed in Africa. And that's to speak to the chevrotains that are around today. So in 55 million years ago, their ancestors were everywhere, I believe. Okay. Yeah, they're interesting little guys. This one is, well, I'm sure this one is no longer alive, but there were chevrotains. This is a photo from, I think, 2005. I had to ask a colleague to get a photo. That's how difficult it is to find anything about them. This chevrotain was at the Bronx Zoo, I believe, and I think they might still have them. I'm not sure, but they're very difficult to find. Okay. Okay, so those are ruminant foregut fermenters. There are other foregut fermenters that are not ruminants. Just like the ruminants, they have an enlarged forestomach. That's the reason why we call them foregut fermenters. Some of them look a lot like ruminants. The hippopotamus has pouches. So if you look at the hippopotamus forestomach, if you don't have your glasses on, that almost looks like a rumen. The forestomach can look different, though. It can be tubular, like in the kangaroo. So in the kangaroo, we have the enlarged forestomach. It almost looks like its cecum and colon is put before the small intestine. One similarity between these different foregut fermenters is that the last compartment is a glandular stomach. You always see that. Every mammal has a glandular stomach. And just like in ruminants, we can have some development of the hindgut. The hippopotamus actually has very little. The kangaroo, it has a small cecum and colon. So yes, we call them foregut fermenters. Most of the fermentation occurs in the foregut, but there is some in the hindgut. So again, it's a spectrum. Okay. So we'll shift to the hindgut fermenters. We'll begin with the cecal hindgut fermenters. And what we see is they have a simple stomach. just like the carnivores, frugivores, and insectivores. Microbial fermentation is not happening there. They have a very large cecum, though. We can see the cecum in the rabbit and the koala. The cecum and part of the colon is very large. This is the site of microbial fermentation. So it's good that we had the question about retention of particles earlier, because the situation is a little different in these animals. They selectively retain fluid and they selectively retain particles, but only fine particles. The fluid and the fine particles are non-fibrous and highly digestible. So this is what contains the sugars, soluble proteins, starches. They will selectively excrete large particles. Those large particles are the ones that are fibrous and poorly digestible. So it's different from the ruminant. The ruminant is retaining large particles, which are fibrous and poorly digestible. In these animals, those are selectively excreted. And we're going to explain why that is just in a moment. Okay, so the reason for this difference is that it permits small body size. So if you see an herbivore that's less than 50 kilograms, it's almost certainly a cecal hindgut fermenter. There are a few other herbivores at the small body size. The explanation is a little tricky. It goes to metabolic rate. So metabolic rate is very high for small body sizes. A rabbit has a much higher metabolic rate per gram of tissue than a horse. And the reason for it, you may have learned it during physiology classes, metabolic rate scales with body mass to the 3/4 power. So again, it's relatively high in small animals, relatively low in large animals. And this poses a problem for small animals that eat an herbivorous diet. Grass, roughage, and leaves is generally poor quality. It's hard to sustain a rabbit-sized animal on fiber because it's slow to digest and sometimes poorly digestible. What retention of fine particles and fluid does is it improves the diet quality and it makes herbivory possible. The rabbit is selecting the best part of the diet, the sugars, soluble proteins, and it is fermenting that. It is fermenting the fiber and the large particles to an extent, but it's really selecting for the better parts of the diet. And that's what makes herbivory possible in this small animal.
I have a quick question.
Yes.
I know that rabbits and other herbivores are considered cecotroph, or where they consume their own dung. Is that called cecotrophs? Is that another highly used method in cecal hindgut fermenters?
Exactly. Yeah, we were not going to mention that, but yes, that is a natural extension of our discussion. So they practice copophagy. They eat their own feces. So another way of dealing with this problem is if you can't digest it the first time, you eat feces and you have another go at it. And so this is a way that the rabbit is coping with herbivory at a small body size. You're absolutely right. Okay, any more questions or comments? That was a good comment.
So how do herbivores digest proteins? If you were to feed a rabbit an egg, how would it digest that? Or is that possible?
So can a rabbit live on eggs? Is that your question, basically?
Yeah, kind of.
Simplified, yes. I know I'm taking it to an extreme. Okay, so they would digest proteins just like, if they're available, they would digest it just like us and just like carnivores, omnivores, animals in which microbial fermentation is not as important. So if the proteins are highly available, like an egg, you know, digestion would start in the stomach and it would continue in the small intestine and be absorbed there. The important thing is that the proteins have to be available. And what I mean by that is they can't be locked away in the cell wall. So if you were feeding them a high quality protein like an egg, they would do just fine. So I think you could actually keep rabbits alive on eggs. It's just if the protein is locked away in the cell wall, then it's not accessible by mammalian enzymes and they would not be able to digest it until they would practice copophagy. So microbes would have access to the protein and they would synthesize their own protein. And then after eating the feces, it would re-enter the stomach and they would have another go at it. So copophagy is particularly important. It's important for fiber digestion. It's important for protein digestion because they're able to get microbes, the protein and microbes that way. So that's a long answer to your question.
Thank you.
Okay. Yeah.
I think the koala is the exception in terms of metabolic rate because it requires less energy than rabbits. They don't move so much.
So right, koalas have a relatively low metabolic rate for their size, and that's because they're marsupials. So I guess you would have to do the math. They definitely have a higher metabolic rate than, say, a kangaroo per body weight. You'd have to do the math to see, you know, where they fall in comparison to other mammals. But right, yeah, koalas do have a low metabolic rate because they're marsupials.
Thank you.
Yeah. Okay, so those are the cecal hindgut fermenters. These are the herbivores that are at small body size. We have other hindgut fermenters, the colonic and cecal colonic fermenters. Okay, so there are some similarities. First, they have simple stomach. That's why we call them hindgut fermenters. In these, they have an enlarged colon. I need to draw it better, sorry. In the pony and African elephant alike, they have an enlarged colon, and this is the site of microbial fermentation. There's also a variably sized cecum. The pony has a pretty big cecum. The African elephant does not. So this pony would be a cecal colonic fermenter, and the African elephant would be a colonic fermenter. The point is that they have a large colon. Unlike the cecal fermenters, there is no selective retention of fluid. The retention of fluid is the same, or it's even shorter than particles. So it's kind of like the ruminant. And these tend to be large. So the pony is large and the African elephant is very large. And they can be large because their metabolic rate is relatively low for their body size. If they had a high metabolic body weight, then they would have to be cecal fermenters because they are retaining particles. They are not selectively excreting the large particles, and so what they're trying to digest is relatively poor quality. Okay? So to try to put everything together and to try to understand why we have so many kinds of herbivores, we'll explain the advantages of foregut versus hindgut fermentation. So there are different situations in which foregut and hindgut fermentation could be advantageous. Foregut fermentation, the advantage here is high efficiency. Foregut fermenters can make a lot out of very little food. there's more extensive digestion because there's longer retention of digesta in the rumen versus the hindgut, particularly when you're comparing, or rumen or other foregut compartments, particularly when you're comparing the foregut fermenters to cecal fermenters. Another reason why they're more efficient, not only do they have more extensive digestion, but they're able to use the products of microbial fermentation more extensively. Microbial products are digested and absorbed in the small intestine. So microbial protein and vitamins, those are absorbed in the small intestine in foregut fermenters. In hindgut fermenters, these are produced in the hindgut and they're not absorbed from there. And so they would be lost to the animal. And the one exception is with animals like rabbits that practice capophagy. They ingest their feces so that they can get the microbial protein and vitamins.
So the foregut fermenters have more extensive digestion because of the time that the food particles are inside the body.
Yes, in the rumen. So in the rumen, particles are digested they're retained much longer than they are in the hindgut for hindgut fermenters.
Okay, thanks.
Yeah. So foregut fermentation is advantageous when animals need to be highly efficient. Hindgut fermentation is advantageous when there is plenty of food available and the animal can eat a lot. So it's good in a high throughput situation. So hindgut fermenters, they can have very high intakes. So the forestomach and foregut fermenters is a site where digested can be digested extensively, but it acts as a bottleneck because of long digester retention in its own site, in the site. So it's a double-edged sword. You retain digesta for a long time in the foregut. That allows it to be digested extensively, but it also acts as a bottleneck and restricts animals from eating a lot. Hindgut fermenters have no such restriction. So think about horses and cows. Horses have a really high intake. They don't have a rumen. They don't have that bottleneck. And so they're able to eat a lot of poor quality feed. Cows, they have a rumen. They will eat a lot less, though they can digest it more extensively. Okay, so that's the comparison between foregut and hindgut fermenters, and it might explain why we see such a diversity in herbivores. Okay, so to try to wrap up everything, these are the take-home messages. The gastrointestinal tract is inhabited by a diverse array of microbes. We'll discuss more about this in the coming lectures, but briefly, it's bacteria, protozoa, fungi, viruses, and methanogens. Microbial growth in the gastrointestinal tract is possible only when the host allows it. Temperature, pH, oxygen concentration, and other conditions must match microbial requirements. Carnivores, frugivores, and insectivores have the simplest gastrointestinal tracts, and herbivores have the most complex. And this reflects the importance of microbial fermentation in these two types of animals. Foregut and hindgut fermenters have different digestive strategies that bring unique advantages, and that may explain why we have both hindgut and foregut fermenters. Okay, so that's it for this lecture. What questions do you guys have?
I have one question.
Sure.
And for the horses, do they need microbial proteins?
Do horses need microbial protein? So they would benefit from microbial protein, but no, they don't have it available to them. Microbial protein is digested and absorbed in the small intestine, but in horses, it's produced in the cecum and colon, and so it's lost to them. I mean, it's found in their feces. So they are able to survive through the small amount of protein that they're able to digest and absorb from the feed using mammalian enzymes. It's always puzzled me how they are able to survive on feed with relatively poor quality protein, but they can do it.
I think so.
I have a quick clarification question with the
Yep.
African elephant.
Yes.
So in that case, based on my understanding, the reason they can survive and persist is due to the metabolic rate. Is that correct?
Right. So their metabolic rate is very low given their body size.
Okay.
It's very low given their body size, and so they can use colonic gut fermentation as an effective strategy. Colonic gut fermentation relies on digestion of large particles, and that would not work in a small animal with high metabolic rate like a rabbit, but it works for large animals like African elephants with very low metabolic rates given their body size.
Okay, thank you.
Yeah, sure.
I also have a question.
Yes.
Do any of the other herbivorous groups besides ruminants chew their cud?
Do any of the other groups besides ruminants chew their cud? I used to know the answer to that one. I believe that kangaroos do, but don't quote me on that. I know that some do, but don't quote me on it. Yes. Yes.
Thank you.
Yeah. Any more questions for clarification?
I have a question about the exams. It's not on the lecture. So if anyone else has a question about lecture, they can go first.
Anything is fair game.
Okay, so for the exams, are they going to be like open note and computer, or are we expected to like put everything away and just go on the trust system that we're not using any materials from the class?
That is a very good question, and I don't have an answer for it right now. I'm still trying to figure out. What you should anticipate is probably closed note. So what you said, the trust system, that's probably what we're going to use. I'm looking at different ways of proctoring, and I'm not finding anything good right now.
Okay, thanks. Just to know how in-depth our notes should be and what we should be studying in advance. Thank you.
Yeah. And we'll have to see. It's going to be a learning experience, so we'll have to see how the first exam goes, and we may adjust things afterwards. And if you're in a graduate class, you're here because you want to learn material, and I'm not going to slam you, particularly under circumstances like these. So, you know, we need to be able to adapt.
Thank you.
Yeah. Any more questions about the exam, about the lectures, about anything in the course? Any concerns that you have?
I have a question about the homework assignment that's due. So you said that our homework will be due on Thursday night.
Yes.
Is there like, do you want us to answer those? Can it be answered like in bullet points, like simplified?
If your answer is clear, you can answer it however you want.
Okay.
So that works. Yeah.
Okay, great. And then do you want us to just submit it as in like a Word file, or do you want us to submit it as like a PDF file? Doesn't matter.
However you want to do it. Right. Yeah.
Okay.
So in the past, most people have submitted as Word files, but if you want to print it off and handwrite, that works too.
Okay, great.
And if I can read it and it makes sense, it's good.
Okay, great. Thank you.
Yeah. Okay. And good point. We have our first assignment due Thursday. It should be available, and you should be able to submit it online. If you have any trouble accessing it or submitting it, just let me know. Okay?
Thank you.
Thank you.
Okay.
Thank
and
you.
We'll see everyone tomorrow. Tomorrow, we are going to begin discussion of microbes of the rumen and discussion of the bacteria in particular. Just as a forewarning, it's probably the most difficult lecture in the entire course. That's one reason why we're doing it early. So please just look ahead and be prepared. There's going to be a lot of memorization involved.
You mean Thursday, not Wednesday?
Thursday, yes. Thank you. My daughter's screaming in the background if you heard that, so I'm a little distracted right now. Okay, yep, thank you.
Thank you.
Okay, yeah, see everyone on Thursday.
Thank you.
Thank you.
Thanks. Okay, bye.
Bye.